Hi friends, happy learning with biology exams for you. In this video, we have combined all core concept videos on photosynthesis for a clear-cut sequential understanding of the topic within 45 minutes. At the end of the discussion, you will be able to understand the equation of photosynthesis that is followed by the site of photosynthesis exactly where the reaction is happening, that is followed by the pigments in photosynthesis, then light-dependent reaction, and also the steps involved in Calvin cycle or light-independent reaction, and the difference between C3 cycle and C4 cycle, and finally, we'll be winding up with CAM cycle. You can use the timestamps to move directly into specific topics. Let's begin with the equation of photosynthesis. So let's start with the definition of photosynthesis. Photosynthesis is an amazing process that is responsible for life on this planet. The reactants are water that is absorbed by the roots, plus the carbon dioxide from the atmosphere. Sunlight is a source of energy, and leaves are the site of photosynthesis. Inside the leaf there is a pigment called chlorophyll that trap the light energy and convert it into chemical energy as glucose, and this is directly or indirectly utilized by all organisms on earth except some microbes. And oxygen is released as byproduct. So this is the overall equation of photosynthesis. 6 molecules of carbon dioxide plus 12 molecules of water in the presence of sunlight and chlorophyll give rise to glucose, water and oxygen. Let us see how we get this equation. Till 1940, this was the equation. 6 carbon dioxide plus 6 water molecule give rise to glucose plus oxygen. It is believed that photosynthesis is just a reverse of respiration. In the case of respiration, this glucose molecule is broken down into carbon dioxide and water with the release of energy. So photosynthesis is a synthetic process, whereas respiration is a catabolic process or breakdown process releasing energy. But this equation doesn't tell much about what is actually happening. What is the exact source of oxygen evolved? Is it from water or is it from carbon dioxide? So this is the experiment that solved the question. Rubin et al. In 1940 used radioisotopes to find out the exact source of oxygen, and this was a paper published. And this was the experiment. In the experiment 1, they used radioactive labeled water as a reactant and found out that the oxygen released is also radioactively labeled. And this was the equation: carbon dioxide plus radioactive labeled water, and oxygen evolved is also radioactively labeled, indicating that this oxygen is formed from water. For the confirmation, they did a second experiment using radioactively labeled carbon dioxide. Here they used radioactively labeled carbon dioxide and found out that the oxygen released is not radioactively labeled, indicating that this oxygen is evolved by the splitting of water molecule. They got water in the product also, which is radioactively labeled. Thus, Rubin et al. Confirmed that oxygen evolved during photosynthesis comes from water. Now we know that it is by the photolysis of water. So these are the two experiments, summary of the two experiments. Now we need to change the equation. Now we know that oxygen evolved during photosynthesis comes from water. Let us take the old equation. 6 carbon dioxide molecule plus 6 water molecule gives C6H12O6, that is glucose, plus 6 oxygen. Let us see what is happening. So now we know that water splitting gives rise to oxygen. Number of oxygen is 6. Here the number of oxygen required is 12. So we need to rearrange this equation. We should balance this equation once again. So this is a balanced overall equation of photosynthesis. 6 carbon dioxide molecule plus 12 water molecule in the presence of light and chlorophyll give rise to glucose, that is C6H12O6, plus 6 water molecule plus 6 oxygen. Let us see, this is 12 H2O. So we have 12 oxygen. Here also we have 12 oxygen. So this is balanced. Now let us check the complete equation is balanced or not. Number of carbon, it is 6. Number of oxygen, 6 into 2, that is 12, plus 12, that is 24 oxygen molecule in the reactant side. Number of hydrogen, 12 into 2, that is 24. In the product side, number of carbon, it is of glucose, that is 6. Number of oxygen, here it is 6. Here also 6. 6 plus 6 into 2, that is 12. 24 oxygen molecule. And number of hydrogen, here it is 12 plus 6 into 2, that is 12. That is 24. The equation is balanced. Now the correct balanced overall equation of photosynthesis is 6 carbon dioxide plus 12 H2O give rise to C6H12O6 plus 6 H2O plus 6 O2, where this O2 is derived from water. Now we know the process. The process is called as a photolysis of water. Hopefully we'll be discussing that in our later videos. Which is a primary site of photosynthesis? It is undoubtedly the leaf. Leaf is the primary site of photosynthesis. But in the case of xerophytes, they may not be having leaf. Then the stem is green. All green parts of the plant are capable of photosynthesis. Leaf is an organ that is designed for photosynthesis. The function of leaf is to absorb light energy and also carbon dioxide. These are some of the adaptations of leaf. There is large surface area to absorb more light. It's very thin so that the diffusion of carbon dioxide is very easy and the distance traveled by carbon dioxide is very short. Then there is an opening called stomata that allows the carbon dioxide entry and also the removal of water vapor by transpiration. And there is a vasculature that allows the translocation of water from root to the site of photosynthesis, that is the leaf. Apart from that, there is a cuticular upper epidermis, and this epidermal layer is transparent that allows the light to penetrate into the mesophyll cell directly, which is a prominent site of photosynthesis. Next question, inside the leaf, which is a cell that is primarily involved in photosynthesis? Leaf anatomy shows that there is an upper epidermis that is followed by palisade layer, then there is a spongy mesophyll layer and lower epidermis with stomata. Mesophyll cells, the stacked cells that is seen just below the epidermis, is a site of photosynthesis. Nearly 80% of photosynthesis occurs in these cells. These cells are abundant in chloroplast. And you can see these cells are rich in chloroplast. So it's a primary site of photosynthesis. Now inside the mesophyll cell, there is a wonderful organelle which is called as chloroplast that is responsible for photosynthesis. This organelle contains a pigment that is capable of trapping light energy and capable of converting that light energy to chemical energy and finally converting it into glucose. This is a structure of chloroplast. It is a double membrane bound organelle, and you can see there are membranous sacs that is stacked one above the other, which is called as granum. Individual membranous sacs are called as thylakoid, and the fluid filled matrix is called as a stroma. Now let us see what is the exact location of each reaction in photosynthesis: light dependent reaction, light independent reaction, and also the photolysis of water. So this is the structure of chloroplast, and you can see this is the thylakoid membrane that is stacked one above the other. So let us zoom in. So this is the thylakoid membrane, and on zooming in, this is the thylakoid membrane. Thylakoid membrane is a place where light reaction occurs, or light dependent reaction takes place in the thylakoid membrane. Photosystem 1 and photosystem 2, all other proteins and electron carriers are located on the thylakoid membrane. Therefore, the exact location of light reaction is the thylakoid membrane. By this process, the energy of sunlight is trapped and is converted to chemical energy as ATP and NADPH. In non-cyclic photophosphorylation, the electrons should be continuously refilled, and there is a process which is called as photolysis or splitting of water that occurs in the thylakoid lumen. Thylakoid membrane is having a space which is called as thylakoid lumen. So the site of water splitting or photolysis of water is the thylakoid lumen, which will refill the electron to the photosystem 2 and also provide H+, and that is utilized for creating a gradient for ATP synthesis. You just see the orientation of ATP synthesis. After light reaction, the light energy is converted to ATP and NADPH. This ATP synthesis is oriented towards stroma. So for Calvin cycle, now the energy is right here in the stroma as NADPH and ATP. Next is the Calvin cycle, where carbon dioxide is converted to carbohydrate. The site of Calvin cycle or light independent reaction is the stroma, where ATP is already there, that is synthesized by light dependent reaction, and also NADPH is also there, that is utilized for the reduction of carbon dioxide to carbohydrate. So this is a summary. In photosynthesis there are two major reactions. There is light dependent reaction and also light independent reaction. Light dependent reaction takes place in the grana or thylakoid membrane of the chloroplast, where light energy is trapped and converted to chemical energy in the form of ATP and NADPH. Whereas light independent reaction, that is the second step, it is also called as dark reaction. It takes place in the stroma, where the ATP and NADPH that is synthesized in the light reaction is utilized to convert carbon dioxide to carbohydrate. And we'll be finding answers to the following questions. What are the pigments in photosynthesis? What is the site of pigments? Three major classes of pigments and the characteristics of each pigment in detail. First of all, starting with the basics of pigments in photosynthesis. Pigments are chemicals that can absorb light in the visible region. This is a visible region, that is from 400 to 700 nanometer. And these pigments include chlorophyll, carotenoids, xanthophylls, phycoerythrin, and phycocyanin. Chlorophyll is the most predominant pigment, that is why these leaves appear green. These pigments are located on an organelle which is called as the chloroplast in leaf. Inside the chloroplast there is thylakoid. This stack is called as granum and each unit is called as thylakoid, and this is an enlarged view of this thylakoid. This is a thylakoid membrane. So pigments are actually located on the thylakoid membrane. You can see for as photosystem 2 and photosystem 1, these pigments are located, and these pigments can absorb light energy and convert it into chemical energy as ATP and NADPH. We call it as light dependent reaction of photosynthesis. So these are amazing chemicals capable of converting the light energy from the sun and converting it into chemical energy as ATP and NADPH that is utilized for the formation of glucose in dark reaction or light independent reaction. Now moving into the details of different types of photosynthetic pigments. There are principal pigment which includes chlorophyll A that is present in all plants and also bacteriochlorophyll that is present in bacteria. Accessory pigments include chlorophyll B, C, D, etc. Carotenoids, which is somewhat yellow or orange red in color, that includes carotene and xanthophyll. Then phycobilins, the third class, that includes phycoerythrin and phycocyanin. Moving into the detail of each pigment. Chlorophyll A. Chlorophyll A is a primary pigment in all plants. Chlorophyll A, the structure of chlorophyll A, you can see there is a central magnesium that is surrounded by 4 nitrogen atoms, and all together this structure is called as a tetrapyrrole or porphyrin head. The formula is 6C55H72O5N4Mg. This is a hydrophilic pyrrole head, and this is the phytol tail which is hydrophobic, that is embedded in the thylakoid membrane. And these 1, 2, 3, 4, these rings are joined by methyl group, and maximum absorption is in the red and blue region and reflects green light. That's why the leaves appear green. It's bluish green pigment. And the side group at the second ring, this is a second ring, the side group at the second ring is a methyl group, that is a CH3 group. Second principal pigment, that is bacteriochlorophyll. In bacteriochlorophyll, which is primary pigment in green and purple sulfur bacteria, the formula is C55H74O6N4Mg. It's a reddish purple pigment with the side group in the CH3 position. It is having CH3C double bond O. In the first ring, the maximum absorption is in the infrared region, that is greater than 720 nanometer. Now accessory pigments. Accessory pigments, first one is chlorophyll B, that is present in blue-green algae and also in all green plants. Chlorophyll B is same as that of the chlorophyll A, except that in the second ring, the side group, instead of methyl group in chlorophyll A, it is CHO group in chlorophyll B. The formula is C55H70O6N4Mg. The rest is the same. Maximum absorption at red and blue region and reflects green light. The only difference from chlorophyll A and B is in chlorophyll B, the side group in the second region, in the second ring, the side group is a CHO group, rather than a CH3 group as in chlorophyll A. Now the second class of pigments, that is auxiliary pigment, that is the carotenoids. It includes carotene and xanthophyll. Carotenoids are auxiliary pigments in all plants. Beta carotene is an example. C40H56 is a formula. Red or orange colored hydrocarbons, maximum absorption in the blue violet region. Whereas xanthophylls are oxygenated carotenes. It can be called as oxygenated carotenes. Example is lutein C40H56O2. You can see that this is oxygenated. Brown or yellow colored oxygenated hydrocarbons. These are responsible for the color of autumn leaves. This is a yellow color of xanthophyll and reddish orange color of carotenoids. As the leaf matures, the chlorophyll deteriorates and is replaced by these auxiliary pigments, carotenoids and xanthophylls. That is why ripened leaf appear as orange or yellowish. Next class of pigments is the phycobilins, that include phycoerythrin and phycocyanin. And this is the structure. Phycoerythrin, these are auxiliary pigments in red algae and cyanobacteria or blue-green algae. It's water-soluble pigment, and this is Volvox, that is a blue-green algae. And also in red algae, this is laminaria. This is red colored and maximum absorption dim and blue-green light. Whereas phycocyanin is a bluish pigment that is present in red algae and blue-green algae. It's also water-soluble. It's blue-colored and maximum absorption. It's extra orange and red light, and that these pigments enables the algae to live in deep underwaters of sea due to the property of these pigments. And this is a summary of what we have discussed. We are going to discuss about what are photosystems, what are the major differences between photosystem 1 and photosystem 2 in detail. Inside the chloroplast, there are membranous sacs that is stacked one above the other, which is called as cranum, and this is a cranum, and which is a site of light reaction. So this is a thylakoid membrane. Let us zoom in this region. So this is a thylakoid membrane. On the thylakoid membrane, photosystems and all other associated electron carriers and proteins are located, or thylakoid membrane is a site of light-dependent reaction. Photosystems are also located on the thylakoid membrane. These are the photosystems, photosystem 2 and photosystem 1. And this is a picture of non-cyclic photophosphorylation. So photosystems are light harvesting complexes that is made up of pigment molecules, auxiliary pigments and associated proteins. Photosystems are located on the thylakoid membrane of the chloroplast in the case of green plants and algae, whereas in the case of bacterium, these photosystems are located on the cell membrane. Let us zoom in this region. Each photosystem consists of two closely linked components, that is a reaction center chlorophyll A molecule and accessory pigment, which is called as antennae molecules. Auxiliary pigments include chlorophyll B, carotene, xanthophyll, etc. What is happening is this energy of sunlight or photons is received by these auxiliary pigments. These auxiliary pigments transfer this energy to other molecules, to the adjacent pigment molecules, by means of resonance transfer, which is a kind of vibratory transfer mechanism. Ultimately, this energy is channeled into the reaction center chlorophyll A molecule, from where electrons are moved from ground state to the excited state, that is received by electron acceptor, and that moves through different electron carriers, providing energy for the creation of electron gradient and ultimately the synthesis of ATP and NADPH. And we have discussed that part in the video cyclic and non-cyclic photophosphorylation in detail. So in summary, these are the functions of photosystem. It is involved in light absorption, transfer of energy, and ultimately transfer of electrons. Let us move into the difference. First of all, starting with difference in absorption peak. So photosystem 1, maximum absorption is 700 nanometer or far red region of light, or it absorbs longer wavelength of light, and it has an iron sulfur type reaction center and is rich in chlorophyll A. In the case of photosystem 2, maximum absorption peak is at 680 nanometer. It has a quinone type reaction center and is rich in chlorophyll B. Second difference is it's regarding the location inside the chloroplast. There are two regions which is called as uppressed and non-uppressed region in chloroplast. So let's zoom in this region, and this is the granum, and individual units are called as thylakoid membrane. So photosystem 1 in green color is located on the non-uppressed granule region, and this is a non-uppressed granule region, or the region that is exposed to stroma. The region that is exposed to stroma, or it can be called as the outer layers of grana. So photosystem 1 in green color, that is primarily, or that is located on the non-uppressed granule region and also on the stroma lamellae that connects the adjacent grana. Whereas photosystem 2 is located on the uppressed granule region. This is the uppressed region. You can see here there is stacking, or the internal part of the grana. Photosystem 2 is with green and black color, and this is seen on the internal part of granum, or the uppressed region of granum. Difference number 3, that is regarding the role in light reaction. Photosystem 1 that is involved in both cyclic and non-cyclic photophosphorylation. In cyclic photophosphorylation, photosystem 1 contributes to the formation of ATP. Electrons returns back to photosystem 1, whereas in non-cyclic, electrons are received from photosystem 2 and it forms NADPH. Whereas in the case of photosystem 2, it is only involved in non-cyclic photophosphorylation and also involved in a process called as photolysis of water. As these electrons, when moves from photosystem 2, there is an electron hole. So in order to refill that electron hole, a process occurs that is called as a photolysis of water, or water splitting, providing electrons and protons. It is involved in ATP synthesis. The 4 stages in light-dependent reaction of photosynthesis. So we have divided the entire process into 4 stages for the sake of understanding. Remember, all may be happening at the same time. So these are the stages: photoexcitation, photolysis, photophosphorylation, and photoreduction. And we'll be discussing these within 3 to 5 minutes. Starting with the first stage, that is the photoexcitation of chlorophyll molecule or pigment systems. So this is a thylakoid membrane where photosystems and all other electron carriers are located along with ATP synthase. Or thylakoid membrane is a site of light-dependent reaction of photosynthesis. So this is a chloroplast stroma, and this is a thylakoid lumen or thylakoid space. So the first step is the photoexcitation of pigment systems. So that this is a pigment system 2 and this is pigment system 1, and this is a picture of non-cyclic photophosphorylation that is predominant in higher plants. Let's see what are photosystems. So photosystems are light harvesting complexes that consist of accessory pigments and a reaction center chlorophyll A molecule. Light energy is trapped by these accessory pigments, and this energy is transferred to adjacent pigment molecules by means of resonance transfer or a kind of vibratory transfer, and ultimately this energy is passed on to the reaction center chlorophyll molecule, and from where the electrons are ejected or raised to the excited state from the ground state, and that is received by electron acceptors, and that will be passed on to other acceptors. Okay, so photosystems are the light harvesting complexes in the light dependent reaction of photosynthesis. Now the electrons are ejected from the photosystem 2. There is an electron hole or an electron gap in photosystem 2 that should be refilled. Then there occurs a process which is called as photolysis of water that occurs in the thylakoid lumen, where the water molecule splits up to form protons and electrons with the release of oxygen. This protons refills this photosystem 2. Protons helps in creating gradient in the thylakoid lumen, and oxygen that is released, and that is the oxygen that is released during photosynthesis. So the second step, stage 2, is a photolysis of water in thylakoid lumen that refills the lost electron in photosystem and also contribute in creating proton gradient. Stage 3 is a photophosphorylation of ADP to ATP. Now there is a proton gradient that is created by the photolysis of water, and also during the electron transfer the energy is utilized to pump protons from the chloroplast stromal region into the thylakoid lumen space. Now here there is more protons compared to this stromal region. So there is no way out for these protons to this side to create an equilibrium. These thylakoid membranes are impermeable. The only way out is a protein which is called as ATP synthase. When the protein moves through this ATP synthase, the energy is utilized for the catalytic activity or for adding phosphate into ADP forming ATP. And this is called as chemiosmotic hypothesis, and this force is called as proton motive force. So simply this proton gradient is created by photolysis of water and also by the pumping of protons from the chloroplastomal region into the thylakoid lumen region, utilizing the energy of electron transfer or electron flow. So while this H plus moves through ATP synthase, the only way for H plus to move out of thylakoid lumen, that energy is utilized for combining this ADP to PI, forming the ATP or the energy rich molecule. And that is the third step. And the final step is the photoreduction of NADP to NADPH. As the electrons moves from photosystem 2, then moves to plastoquinone, B6F, which is a proton channel, then plastocyanin, and that is received by photosystem 1. This is further energized by the sunlight. Then there is a ferredoxin reductase enzyme that is close to photosystem 1, and that will reduce NADP in the chloroplastomal region to NADPH. So at the end of light dependent reaction, the energy of sunlight that is received by the photosystems or chlorophyll molecule is converted into chemical energy in the form of ATP and NADPH. These two energy rich molecules, that is ATP and NADPH, will be used in converting carbon dioxide or CO2 to glucose C6H12O6 in Calvin cycle or C3 cycle. And we'll be discussing that in the next video. And that's it. These are the 4 stages of light dependent reaction of photosynthesis. Remember, all these things may be happening simultaneously. I think it is better to understand Calvin cycle by knowing the exact reaction that is happening. Calvin cycle is also called as C3 cycle, Calvin-Benson-Besham cycle or CBB cycle, reductive bendose phosphate cycle. Let's begin. Starting with the reactions. The first reaction is carbon fixation, where carbon dioxide combines with RUBP, ribulose biphosphate, in the presence of enzyme Rubisco, forming a 3-carbon compound. First, there is a formation of a short-lived 6-carbon compound, then that splits to form a 3-carbon compound, which is 3-phosphoglyceric acid. Therefore, the cycle is also called as C3 cycle. Step number 1 is carbon fixation, where carbon dioxide combines with RUBP, forming a stable 3-phosphoglyceric acid. Step 2 is phosphorylation. This step is added for better understanding so that what is happening in these reactions. This 3-phosphoglyceric acid is converted to 1,3-biphosphoglyceric acid. A phosphate group is added, therefore ATP is required. Therefore, in this step, ATP is utilized. Step 3 is reduction. In reduction, this 1,3-biphosphoglyceric acid is converted to glyceraldehyde 3-phosphate. Acid is converted to aldehyde, where NADPH is reduced to NADP. And this glyceraldehyde 3-phosphate, which is a very common metabolite inside the cell, is used to synthesize glucose. 2 glyceraldehyde 3-phosphate molecules are used to make a glucose molecule. Step 4 is glucose synthesis. 2 glyceraldehyde 3-phosphate molecule, as it is 3-carbon, is used to make a glucose molecule. And the step 5 is regeneration of RUBP from this glyceraldehyde 3-phosphate. Here also ATP is utilized for regenerating RUBP. Now let us see the exact reactions with numbers. 6 carbon dioxide molecules combines with 6 RUBP. Now we have 36 carbon, 6 carbon plus 5 into 6, that is 30, 30 plus 6, 36 carbon atom in both these compounds. to form 3-phosphoglycerate. Therefore, this is a 3-carbon compound. Therefore, 36 by 3, 12 phosphoglyceric acid will be formed. Then this 12 phosphoglyceric acid, then it is converted to 1,3-pi-phosphoglycerate or glyceric acid. Whenever there is an addition of phosphate, the enzyme is kinase. Here the enzyme is phosphoglycerokinase. This is the substrate name. Here 12 ATP is used as 12 1,3-pi-phosphoglycerate is formed. Then in the reduction stage, the enzyme is dehydrogenase, the triose phosphate dehydrogenase. 12 NADPH is used in this step. Then 12 glyceraldehyde 3-phosphate is formed. Out of this 12 glyceraldehyde 3-phosphate, 2 glyceraldehyde 3-phosphate is used to make 1 glucose molecule. As glucose is 6 carbon, 2 glyceraldehyde 3-phosphate molecule is required. Now we have 10 glyceraldehyde 3-phosphate molecule, and that is used for regeneration of RUBP. 10 glyceraldehyde 3-phosphate, that is 30 carbon, and it will form 30 by 5. RUBP is having 5 carbon, therefore it will regenerate to form 6 RUBP, thus completing the cycle. Here also 6 ATP is utilized for regeneration. So in total 18 ATP is utilized for synthesis of a glucose molecule, where 12 ATP is utilized at the beginning for phosphorylation, then 6 ATP for regeneration, and 12 NADPH is utilized for the synthesis of a glucose molecule. So this is 2 glyceraldehyde 3-phosphate molecule forming a glucose molecule which is having 6 carbon. It's better to understand by remembering the exact reaction than using some mnemonics or abbreviations. Let me repeat once more. RUBP combines with carbon dioxide, forming the first stable compound is 3-phosphoglyceric acid. Then it is phosphorylated to form 1,3-biphosphoglyceric acid. Then it is reduced to glyceraldehyde 3-phosphate. 2 molecules of glyceraldehyde 3-phosphate makes a glucose, and the rest is used for regeneration of RUBP. The enzymes involved in regeneration are transketolase, ribose phosphate isomerase, phosphoribokinase. The difference between Calvin cycle and C4 cycle, or C3 cycle versus C4 cycle. So why C3, C4 and CAM? C3 is a normal cycle. What's the reason for certain plants exhibiting C4 and CAM cycle? The first and the most important thing to remember is all plants makes glucose by C3 cycle or Calvin cycle, and it is a default cycle for the synthesis of glucose. Calvin cycle is a cyclic reaction involved in the synthesis of glucose from carbon dioxide with the use of ATP and NADPH that is synthesized during the light dependent reaction of photosynthesis. We have discussed that in detail in the video regarding 4 stages of light dependent reaction of photosynthesis. Now C4 cycle, actually it is an adaptation to survive in dry habitats for 2 reasons: to minimize transpiration and also to nullify photorespiration, a process that involves wastage of energy, thereby increasing the photosynthetic efficiency. CAM cycle, it's an adaptation to live in desert condition, and we'll be discussing that in the next video. So the most important thing is C3 cycle is the cycle that is in all plants that is responsible for the synthesis of glucose. Wheat is a C3 plant and maize is a C4 plant. Now moving into the differences. First difference number 1, why C4 cycle? Primary carbon dioxide acceptor and the enzyme involved in the first step. Let us discuss about the most important enzyme in the process, that is Rubisco. This is a Calvin cycle. In C3 cycle, carbon dioxide combines with RUBP or ribulose biphosphate, and the enzyme is Rubisco, ribulose 1,5-biphosphate carboxylase oxygenase. It is having carboxylase activity. It can bind to carbon dioxide, and forming a C3 compound, and during the C3 cycle ATP and NADPH that is synthesized in the light reaction is utilized and ultimately forming glucose. This Rubisco is having yet another activity, which is the oxygenase activity. If the concentration of oxygen is high, this Rubisco will bind to oxygen rather than carbon dioxide. On binding of oxygen, Rubisco forms 2-phosphoglycolate, therefore the first stable compound is 2-phosphoglycolate, therefore called a C2 cycle. Then in order to regenerate this 2-phosphoglycolate, a great amount of ATP and NADPH that is synthesized in the light dependent reaction is used, and the net result is simply the release of carbon dioxide. That is why this is called as photorespiration. Just like our respiration, oxygen is taken in and carbon dioxide is released without the production of sugar or glucose. In order to regenerate this C2 compound, phosphoglycolate should pass through 3 organelles: chloroplast, peroxisome and mitochondria, with the expenditure of ATP and NADPH. So this cycle is a wastage of energy with a huge expenditure of energy without having any beneficial aspect. So the plant that is living in dry condition always tries to avoid this photorespiration. That is why there is C4 cycle. Now moving into the differences. And this is a C3 cycle or Calvin cycle. Here the first stable compound RUBP combines with carbon dioxide in the presence of enzyme Rubisco, forming a 6-carbon short-lived intermediate. The first stable compound is 3-phosphoglycerate. Therefore this cycle is called as C3 cycle. And the primary carbon dioxide acceptor is RUBP, ribulose biphosphate, that combines with carbon dioxide, and the enzyme involved is Rubisco. In C4 cycle, carbon dioxide combines with phosphoenol pyruvate or PEP, forming oxaloacetic acid, and it's a C4 compound. You can see there are 4 carbons. Therefore this cycle is called as C4 cycle. Here the primary carbon dioxide acceptor in mesophyll cell is PEP or phosphoenol pyruvate, and that is converted to oxaloacetate, a 4-carbon compound, and the enzyme involved is PEP carboxylase. Difference number 2 is regarding the occurrence. C3 cycle, this is common, very common in wheat, potato, soybean, rice, etc. Occur in all plants including C4 and CAM plants. More than 85% of plants are C3, only C3 cycle is going on in such plants. Optimum temperature is 20 to 25 degrees Celsius, and this cycle works well in an environment where there is sufficient water with moderate temperature and sunlight. In that condition, even though there is photorespiration, that will not harm this plant. Whereas C4 cycle is actually an adaptation. Examples include maize, sorghum, sugarcane, millet plants, etc. And it occurs in approximately 900 species. It's an adaptation to reduce photorespiration. Majority are monocots, and these C4 plants can grow well in an environment with limited supply of water or dry habitats and high temperature and high sunlight. So during high temperature and high sunlight, if there is photorespiration, these plants cannot survive. That's why there is a cycle called C4 cycle that will nullify the effect of photorespiration. Now moving into the difference number 3, that is site of carbon dioxide fixation and Calvin cycle. In C3 cycle, initial carbon dioxide fixation and Calvin cycle, everything occurs inside the mesophyll cells. You can see carbon dioxide entering, oxygen is moving out, and water is also released by transpiration. Whereas in C4 cycle, initial step, that is initial carbon dioxide fixation, takes place in the mesophyll cells. Later it is transported into the bundle sheath cell where Calvin cycle occurs. Therefore, initial carbon dioxide fixation and Calvin cycle is separated in space at 2 locations. This picture will tell you the details. Initial carbon dioxide fixation by PEP occurs in the mesophyll cell. Then that is converted to malate. Malate is transported to bundle sheath cell where carbon dioxide is decarboxylated, so that carbon dioxide is made available for Calvin cycle. So in bundle sheath cells, carbon dioxide is accumulated. Bundle sheath cells acts as a carbon dioxide concentrator, providing Rubisco with an option of optimum carbon dioxide concentration, thereby nullifying or completely avoiding photorespiration or the inhibitory effect of oxygen. And later this malate is converted to pyruvate and later converted to phosphoenol pyruvate. Difference number 4 regarding leaf anatomy. There is no crans anatomy in the leaves of C3 plants. There is a single type of chloroplast, vascular bundle, that is surrounded by bundle sheath cell without chloroplast, and the site of photosynthesis is a mesophyll cell. You can see the green color. Whereas in C4 cycle there is a specific anatomy that is designed for this C4 cycle. Vasculature is surrounded by bundle sheath cells with chloroplast, and that is surrounded by mesophyll cells with chloroplast. And mesophyll cells are the site of initial carbon dioxide fixation and light reaction. Therefore, this site is rich in grana. It is having well developed grana, whereas bundle sheath cells are the site of Calvin cycle or light independent reaction or dark reaction. Therefore, this chloroplast is agranal, or grana is poorly developed in bundle sheath cells. Therefore, there are two types of chloroplast: granal in mesophyll cells and agranal in bundle sheath cells. And this anatomy is called as crans anatomy. This 2D picture will give you a better clarity. Vasculature that is surrounded by bundle sheath cells with chloroplast, that is further surrounded by mesophyll cells without much space, or direct contact with the bundle sheath cells without much space. And this anatomy is called as crans anatomy, a bouquet-like arrangement. Difference number 5, that is regarding the photosynthetic efficiency and photorespiration. C3 cycle is a cycle that is meant for plants that is having sufficient water. So photorespiration occurs that reduces the photosynthetic efficiency. 12 NADPH and 18 ATP molecules are required for synthesis of one glucose molecule, and we have discussed in detail in the last video on Calvin cycle. So in the C4 cycle is an adaptation. Thereby the photorespiration is completely nullified or absent. That increases the photosynthetic efficiency, yield up to 50%, approximately 50% more efficient than C3 plant. C4 plants are well adapted to survive in dry habitats. But for transporting all these things, malate from mesophyll cell to bundle sheath cells, then regeneration of this malate to phosphoenol pyruvate, ATP is required. So per glucose molecule, 12 NADPH and 30 ATP molecules are required in the case of C4 cycle. The cycle is a bit expensive as far as ATP is concerned, but can avoid photorespiration, which is much more adverse. And that's it. Interesting topic, that is CAM cycle in plants. CAM cycle is an adaptation of desert plants to survive in water deficient environment. If you are new to this channel, please subscribe and support this channel. Moving into the topic, starting with CAM pathway. So this is a desert plant, and you can see this mesophyll cells of this plant. Suppose this is a mesophyll cell of this plant. The major concern of this plant is to conserve water. So this pathway allows the plants to conserve water. So let us move into the cycle. First step is during night time, stomata open in CAM plants. CAM plants, the stomata is having a speciality which is called as photoactive. It can open during night time and it can close during daytime. That type of stomata is called as photoactive. Stomata opens during night and carbon dioxide enters, and it combines with PEP, forming oxaloacetic acid in the presence of enzyme PEP carboxylase, just like C4 pathway. And that oxaloacetate is converted to malic acid, and that is transported to vacuole, and it is stored during night time. Therefore, the intracellular acidity increases. That is why this phase is called as acidification phase. During daytime, in order to avoid water loss by transpiration, this plant closes its stomata, and malic acid is stored in the vacuole, and that malic acid is taken out, and that is decarboxylated to release carbon dioxide. This carbon dioxide runs the Calvin cycle. It enters the chloroplast and runs the Calvin cycle. And this malate is decarboxylated to pyruvate, and that is recycled back. So the advantage of this pathway is, even without opening the stomata during daytime, Calvin cycle can run using the carbon dioxide that is produced by the decarboxylation of malic acid that is stored in the vacuole during nighttime. So the point in CAM cycle is that initial carbon dioxide fixation occurs during night and Calvin cycle occurs during daytime. And both these are separated in time, and both reactions occurs in mesophyll cells. Here the first stable compound, just like C4 cycle, here also it is oxaloacetic acid, and this pathway ensures minimum photorespiration and prevents water loss by transpiration that allows such plants to survive in desert condition. Now, why C3, C4 and CAM pathway? The first and the most important point is all plants makes glucose by C3 cycle or Calvin cycle. Calvin cycle, as we know, it is a cycle that is involved in the synthesis of glucose from carbon dioxide with the use of ATP and NADPH that is synthesized during light reaction of photosynthesis, and that is the cycle that is universal to plants for the synthesis of glucose. Whereas there is C4 cycle, which is also an adaptation to survive in dry habitats that minimizes the wastage of energy by photorespiration, thus increases the photosynthetic efficiency of the plants. Then the third pathway is a CAM pathway, which is called as cresylation acid metabolism cycle, as this pathway was first observed in the members of family Cressylaceae. It's an adaptation to live in desert condition that ensures minimum water loss by transpiration. Now, examples of CAM plants. Examples include pineapple, agave, cacti, orchid, all are examples. Plants that are adapted to live in hot, dry desert environment. Approximately 8% of all land plants are CAM plants. Majority are succulents. The advantage of CAM plants is photorespiration is very much minimized or suppressed, and transpiration rate is very much reduced as stomata opens only at night time. Hope you understand the concept. Thank you so much for your support. You are with biologyexamsforyou.com.